This method can help answer key questions in the plant development field regarding the distribution of growth regulatory phytohormones.
By using genetically encoded threat biosensors, for example, NLS GPS1, which detects gibberellins, it is possible to measure the dynamic distribution of important compounds in Arabidopsis tissues at a cellular resolution.
Generally, individuals new to this method will struggle because analyzing threat biosensors involves ratiometric imaging, which has increased noise and additional analytical steps compared to intensiometric imaging.
Begin by sowing approximately 20 sterilized Arabidopsis seeds on 1/2 Marashigi and Skug or MS agar square plates. Seal the plates with porous surgical tape and wrap them with aluminum foil for 1 to 3 days of incubation at 4 degrees Celsius for stratification. For root imaging, transfer the plates to a growth chamber in a vertical orientation for 3 days under the indicated conditions. For dark-grown hypocotyl, transfer the plates to the growth chamber for a 1 to 4 hour light pulse to synchronize the germination. Then wrap the plates with aluminum foil and place them into the growth chamber in a vertical orientation for 3 days. For steady-state measurements, add 50 microliters of mock solution to a clean microscope slide and gently place 3 nuclear localized Gibberellin Perception Sensor 1 or NLS GPS1 expressing seedlings onto the slide. Then spot a drop of vacuum grease on each corner of a clean cover slip and gently